Welcome to Vital Science Institute, your one-stop solution to life science. In today's video, we will discuss about histone modifications. We all know the basic structure of histone. Here, this is a histone and histone octamer, which comprises of H2A, H2B, H3 and H4, and there lies DNA that wraps around this histone 1.65 turns. In some of the books you will find 1.75 turns, but when it comes to Watson you will find 1.65 turns. Now the fact is, when it comes to modification, the main purpose of it is gene activation or repression. Now what does it mean? Suppose our DNA is wrapped around the histone. Now this DNA needs to be replicated as well as transcribed for a particular gene to function. For that, the DNA needs unwrapping. Now this kind of unwrapping or rewrapping all around the histone is mediated by this kind of mechanisms like acetylation or methylation. Now let's dive deep into it. When it comes to acetylation, it is majorly associated with gene activation. Now, what does it mean? Suppose among the four histones, suppose we are choosing H3. Now all of these histones, whether it be H2A or H2B or H3 or H4, all of them, they have a N-terminal tail. Now this N-terminal tail, it bears basal amino acids like lysine or arginine. Here we will consider about lysine. Now let's see, suppose this is histone H3. And within this histone H3 lies our N-terminal tail. Among the N-terminal tail lies our lysine residue. We all know the structure of lysine, which bears the R is CH2, 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 and there lies a amino group. Because of this NH3 plus, this entire histone is positively charged. And due to this reason, as we all know that DNA is negatively charged, it remains wrapped around the histone. Now, during this mechanism of acetylation, what happens is, there lies an enzyme called histone acetyl transferase, which transfers the histone, which transfers the acetyl group within the lysine residue. As a result, as you can see, in this transformed approach, here the H is replaced by our acetyl group. Now binding of acetyl group with the lysine residue, binding of acetyl group with the lysine residue, what it does? Let's see. It basically neutralizes the entire histone. Due to this kind of neutralization, what will happen? The DNA won't remain attached with our histone, and finally it will undergo unwrapping. Due to this unwrapping, the kind of activation will occur. Now coming to the fact that when this kind of acetylation of histone occurs, mainly at histone H3, there lies certain proteins or domains that gets attracted towards this acetylated region. These proteins are bromodomains. This bromodomain, what it does, whenever this entire lysine residue gets acetylated, the bromodomain comes and binds with this acetylated region. And this is how the entire mechanism of acetylation leading to gene activation takes place within the histone. This is the first part of our discussion. Now, another kind of modification is methylation. When it comes to methylation, it can be based upon activation or repression. Both of these phenomena can occur. Activation of gene or repression of gene both can occur when it comes to this kind of approach. Now what happens? Let's see. Again we are taking histone H3 as for example, and we have our N-terminal tail. Within that N-terminal tail lies our lysine residue, and we know the structure of lysine bearing a positively charged amino group. Now what happens? There lies a enzyme called histone methyl transferase. Histone methyl transferase. What this enzyme does, it transfers the methyl group within the lysine residue. Now, this transfer may occur in three distinct ways. In one case, only one methyl residue can be added. In another case, two methyl residues can be added. And in the third case, even three methyl residues can replace the H ions of amino group. But this kind of transfer does not neutralize. This kind of transfer does not neutralize the overall structure. And due to this process, what happens? Two distinct mechanisms can simultaneously occur. It can either lead to activation or it can lead to repression. Now, what happens? Suppose methyl group is added. As you can see from this statement I have written, binding of methyl group within lysine residue at specific position. Now, what does this mean? Let's dive deep into it. Suppose we have our histone H3. This is our N-terminal tail, and suppose this is our lysine residue. Now, if we consider the overall orientation of amino acid 1, 2, 3, 4, 5, 6, 7, 8, 9, something like this. Suppose in this pattern, the overall amino acids are being aligned. Now the interesting fact is, when it comes to methylation, there lies specific lysine residues at specific positions that will determine whether that gene will undergo activation or repression. Now what does that mean? As we have seen previously, lysine 9 of histone H3 and lysine 27 of histone H3, wherever this mechanism is kind of occurring, this will lead to our gene repression. This will lead to our gene repression. That means transcription or translation of any of the protein will be simply obliterated when this kind of methylation will occur at specific lysine residues within histone H3. But when it comes to lysine 4 as well as lysine 36 of histone H3, if this kind of methylation occurs, both of them are methylation. If this kind of methylation occurs, then that will lead to gene activation. So what is the mechanism involved? Let's go back to our previous picture. As you can see, lysine residues at specific position will determine whether it will relate to activation or it will relate to our replication. Now what happens whenever this methyl group gets added with the lysine residues at specific sequences or positions, it attracts certain group of proteins. These proteins are called chromodomains. These proteins are called chromodomains. Note it. Cause this chromodomain, what it does? It initiates the mechanism of chromatin remodeling, and this chromatin remodeling will determine whether the methylation is kind of activating or repressing our gene of interest. So when it comes to acetylation, there lies chromodomain at the acetylated site, and when it comes to methylation, there lies our chromodomain that binds to our methylated regions. And this kind of chromodomain binding will determine what kind of activity will be performed, whether it be activation or repression. Now coming to our after effects. Now as you have seen from the picture, histone, there lies acetylation and there lies methylation. Acetylation, when acetylation occurs at histone H3, there lies two distinct positions, lysine 9 and lysine 14. Now what it does? It recruits. How come the activation by means of acetylation is occurring? What it does? It recruits our transcription factor 2D. It recruits our transcription factor 2D. Note it. And this will initiate the mechanism of transcription, followed by gene activation. And when it comes to histone H4, there lies two distinct, there lies histone 8, lysine 8. That means histone 4, lysine 8 at N-terminal region. What it does? Due to this kind of acetylation, it will recruit the chromatin remodeling complexes like SNF. Note it down. So, when it comes to acetylation at histone H3, there lies two distinct residues, one at histone 9, another at histone 14. Now, if this acetylation occurs, then that will lead to recruitment of TF2D, which is a basal transcription factor, that will recruit other transcription factors as well, and thus the transcription in case of eukaryotes will take place. And when it comes to histone H4, there lies our lysine residue, specifically lysine 8. If acetylation occurs at lysine position 8 of histone H4, then what will happen? Then it will recruit the chromatin remodeling complexes like SNF, and hence due to this mechanism the activation must occur. When it comes to inactivation by means of methylation, as we have previously discovered, there lies two distinct phenomena. Activation will take place by means of methylation at lysine 4 and 36, as we have seen over here. Lysine at K4 and K36 will lead to activation, and histone H3, lysine 9 methylation and lysine 27 methylation will lead to our repression. Now what repression? How come this repression is occurring? Repression takes place by means of recruitment of SYN3A protein. Now what this SYN3A protein does? It acts as a co-repressor with eject. Eject, what is eject? Histone deacetylase that removes the acetyl group from the histones. So SYN3A, it acts as a repressor, co-repressor, and and it binds with our eject, and whenever this kind of complex comes into action, then repression must occur, and these two phenomena will occur when methylation will take place at lysine number 9 and lysine number 27. Note it down. The rest of the methylation by means of DNA will be discussed in our next class. If you find this video interesting as well as informative, do share with all of your batchmates. And always remember, stay focused, build concepts and track progress. Me, Shankar Banerjee, founder and educator at Vital Science Institute, signing off.